sample. Okay? And then ask one book to publish, you need 3 replicates, remember. Okay. So now, you take the protein fragment, that's called top-down. You make the peptide, okay, by trypsin and fragment. If you don't have in lab trypsin, also there is way to work. Okay? You can take whole the protein, put the mass spec, that's called native mass spectrometry. Whole mass you are getting, and you can study lot of things. I am not going in the deep. If you have no trypsin, check the charge state, fragment, you can find the mutation and you can go ahead. If you have trypsin, you also fragment, MALDI, ESI, LCMS, any machine, don't worry about it. You will get nicely characterization and then done. You can see that. We can do sickle cell anemia, you can do anything. Any mutation, anything you can carry. Simple way I try to, how can you do? Now see complex. I hope you understood?
Yes, sir.
Raise the hand.
Yes, sir.
Sleeping, everyone is looking WhatsApp and other. Why? You will not get chance. Okay, now is it possible to cancer progression? It's a very difficult thing. How it progress, how it metastasis, how you do the things. There are several cancers we have put, I will not go in detail. We have labeled, select, quantitate, 10,000 protein. Match all these protein, mostly are overlapping. Hand has also 20,000 protein, leg has also 20,000 protein, all both are different. That's the quantity, how it has been arranged, and we are only trying to decipher. You label it, merge 1 cell line, merge 5 cell line, merge 11 cell line, you will get roughly same protein. What it shows, your most developed technology is still not developed. And I can say underdeveloped. Why? The 11 cell line I am getting same. Why we are not getting 20,000 protein? Okay, there is nothing comments. It's the way how far proteomics and instrument has to go. Okay, so one shot you can get 4,000, 5,000. It's enough. Your PhD will be over to study 5,000 protein. Maybe your life will be over to study 5,000. Why to worry? Okay, so move it. Now you see quantity and do this quantitation and you can see figure out. I will not go do in details. You will plot, nice plot for nice figure, for nice paper. This all are important. Why? I will show you now. Now you go anti-cancer, we have compound, we have different way. Melanoma, I will not go in detail, it's a very aggressive cancer. No, okay. And that's okay. Now, a student needs patience here. How? You have to generate the model. You have to generate cancer, you have to generate metastasis, and then you have to study, and then you go to directly therapy. This is the way, here you see, nice poor mice, okay? We have ethical, so we go and we make nice tumor. Sometimes tumor was so big size that, and every day you collect the blood, every day you see, and then try to see how it progressed, and where it gone to metastasis. That's the key, that's the thing. One way I told you, there are several ways to make. We are making 4 or 5 way tumor in the lab. One way I am showing you. The student is Shreya, that's a super patient. Now you have every day go measure and see that how tumor is forming. Now you do the histology. If really tumor is benign or malignant, you can figure out, you can have histology and completely you can do it. That's the way we make the sample, we make the proper and try to understand. Then do histology of different organs. It is not that tumor is only there. It affects all the body, okay? And then different level are there, different days it affects different way. So you can see histology of spleen, where full melanin pigmentation we can get. Now you do proteomics, proteomics symposium. Now you see, when you are studying proteomics, what you have to do different day. Data is completely large. How to narrow down on what is our RAS related. But we are not interested in RAS, because RAS, everyone knows protein is there. What this guy can affect, pancreatitis associated protein, this is almost a death mark. So that means when mouse is going close to die, before that you can guess. So very nice progression is there, and then send me a WD repeat, we are interested in living. So according to your interest, you will jump on the problem. Now, we are trying to repurpose some drug and to find the drug. Chloroquine, I hope you all know, malarial drug, very cheap drug, okay, very low cost, wonderful. We try to repurpose. This is an example. You put in the cell culture, and then you see how it is working very well. You put in the details, take the photographs, and see how it is working. Looks like better. Now, put the detailed study, facts, and other things. Here is that. Cell culture is cell culture, mouse is mouse, human is human. 100 will work in cell culture, 10 will work in mouse, hardly one goes to human. That's the practicality. You have to understand. But here is a nice case. If you don't treat, this tumor size is going up. You give just chloroquine treatment and this never gone up. It took time and here it gone. You see how the patients the student has, they gone to 4 months every day collection, measurement and that. So that's the wonderful, it shows that this compound is working, simple chloroquine, let's see. But remember, this is a mouse study, preclinical, clinical, long way. So nothing to come jump conclusion, this is the research. I'll give you another example. With hernia, I hope you have understood the concept. Now I will go fast. With hernia, with heparin, we are working from this long time. Here, just put the extract, it is working, wonderful. You go to facts, you go do DAPI, you do DNA fragmentation, all these study you do. It looks like you are doing mechanistic and what, some details that's come. But when you do proteomics, what you see? You see here, completely metabolic process is coming very high. So that's way it is going fine. So whole proteomics is giving you. Now you go classical way, put the student asked to run HPLC day and night, only go according to their comfort, and you see characterize the compound. We came to know that this compound was the with heparin A. So that's way we came to know heparin A, and then you see IC50 is going very down, and after that you do detailed study of fats and completely this way. Last, another compound, and I will close with that. Alizarin yellow, this is simple dye. You see how this is working. Now you take all the melanoma drug, put on there, you'll come to know it is working on APT signaling pathway. Now nothing to do, very simple. Do proteomics study and see where usual pathway is going, what is the expected. But again, proteomics is proteomics. Till you will not do Western blot, no journal will accept. So you have to do that also. Validation is there. You have to go in real life. Okay? So now you can do that. So what is the most interesting thing in this last slide? P53, you know, it is the cancer suppression gene. So when cancer comes, this cancer suppression gene will be down. Okay? When you give the treatment, we expect this should go up. And it has gone up. BCL2 is the oncogene, cancer-related gene. And when cancer comes, BCL2 will go up. When you treat it, it should go down. So after treatment, it went down, proved by proteomics, proved by your Western blot, and that shows our concept is working. Let's see how far, how long I will take it. Now, you put in the signaling pathway, you came to know this is AKT signaling pathway. That's the way I showed you how target, how discovery, how biomarker, and how you can take to therapeutics by different way with this. Thank you. I hope you have enjoyed the talk.